So I think that makes 6 questions extra credit. 6 extra credit. So you have 55 questions plus 6 extra credit questions.
Huh.
So it doesn't matter which question is extra credit and which question is a regular question. What I'm going to do is calculate your score over 55 questions. So in other words, I'm going to calculate your grade as if there were only 55 questions. So let's say you got 57 questions right. Well, then you have more than 100. You got extra credit. 57 is more than 55. See? So I'm going to calculate your grade out of 55 questions, as if there were 55, but there's actually 61, so 6 extra credit questions. So that way it doesn't matter which ones you get right and which ones you get wrong, they all count the same. Yeah, they all count the same. It's called hidden extra credit, because I'm not supposed to give extra credit in this class. Yeah, sophomore level, yeah, right? Sophomore level, they're like, no, and that's their last class before nursing, you can't give them extra credit, so I can't have this. So as far as anyone's concerned, there's only 55 questions, but yeah, we get 61. On your lab practical, however, I did put 5 extra credit questions, and they count the same too, but you'll know definitely those 5 are the extra credit questions. But again, it doesn't really matter which ones you get right and which ones you get wrong, because they all count the same. They're all 2 points each. Alrighty, so if you're ready, this should be the easiest final exam because it's a test of your general knowledge. Did you kind of grasp it as we went through the semester? So it's, in most cases, less detailed, more general information, just to see if you got the gist of the whole course. So, okay, so going back to the beginning of the class, do you remember we studied microbiology, and their definition of microbiology was organisms and structures too small to be seen with the naked eye, and so that would be 1/10 of a millimeter or less. And there were 3 things that led to the study of microbiology. What were they?
Microscope.
Right. The development of the microscope, and who was the guy who got credited for using a microscope, drawing pictures of critters and sperm cells, and what was the second thing that led to the study of microbiology? Yes, a controversy over spontaneous generation. Where do these little animalcules come from? And what was the third part?
What caused the disease?
Yeah, what caused the disease and how is it transmitted? Good. And we also studied the foundation for the germ theory of disease. So finally, those 3 things, and learning culturing techniques and growing microbes in broth and whatever, they finally figure out that germs cause disease. And we called that the germ theory of disease. Germs cause disease. But somebody had to show that germs cause disease, and in fact makes a direct correlation between a particular germ and a particular disease. Oh, what kind of critter? Don't want to freak them out.
They only do.
If it's a snake, let's. What is it?
What's the question?
So yeah, get it onto there.
What is it?
So particular bacteria called
It's huge.
What is it?
phastoids.
A roach? I would be jumping down my feet
What
right
was the
now.
question?
Something that you
Oh, it's a tiny little bug.
Yeah, let's not go through that door.
Oh,
Let's not go through that
You
door.
have to take them out over there.
Most of them have things like
You have to go for a walk. It's not a tick, is it?
No, it's not.
Yeah, I don't want Lyme disease. Let me see that critter.
It's a small.
Oh, it's a true bug. I forget what the family name for that true bug. It's a true bug. Yeah, they call them true bugs. It's a true bug. I can't remember what the family name is.
So what was the question what we were talking about?
Okay, so what was the foundation foundation for the germ theory. What do we call that four-step thing, process? Koch's
Postulate.
postulates, right. So Koch's postulates is the foundation for the germ theory of disease. And when Dr. Koch did his postulates, first of all, you've had this on multiple quizzes and exams. What's the hardest part, even before you can do Koch's postulates?
Culture.
You gotta culture that microbe, you know, first you got something. I saw the thing on the news about Legionnaire's disease, and at the end of the news clip, they said, and usually it's elderly or weak people, immunocompromised people that get Legionnaire's disease.
Yeah,
But
it was
still, there was a pretty significant number of people who got it, and, they thought it was from the water cooling tanks or
Well, they
something.
had it from this, sprinkler heads and air conditioning brought on too, and they had 15 children that died from that as well.
What town was that?
New York.
New York or
Oh, yeah, the
Chicago.
Bronx, it was in the Bronx.
Yeah.
Yeah, that's right, right. Well, something that wasn't publicized a lot, I think it was maybe 2 years ago, there was a tuberculosis outbreak in the Bronx because people, these Hasidic Jews don't believe in getting vaccinated. And if they came from wherever they come from in Europe, they normally do get vaccinated, but the Hasidic Jews don't get vaccinated. And the Hasidic Jews all lived in the same building because they're a very tight-knit group, you know? And so one guy with tuberculosis got in an elevator to go down shopping, whatever, and if you cough or sneeze in the elevator, the Mycobacterium tuberculosis germ can actually persist until another person gets in the elevator, and then you're trapped. I mean, it's an elevator, you have no choice, that's the air you're gonna, and they breathed in tuberculosis. So there was like an epidemic in the building. And also, now we've got a cluster of people all leaving the building to do their shopping and go to church and whatever, or not church, temple, or whatever, you know? And so they spread it to other people. But fortunately, the health department was able to link it back to that building. And the people in that building all had tuberculosis. That was like 2 years ago. It was like, that's a scary thing. Hello, New York City, really? You're gonna have people with tuberculosis walking around New York City? They could infect tons of people. Not everybody's susceptible, apparently, but I guess enough people are. Some people have kind of a natural immunity to tuberculosis. Which reminds me, I saw a critter outside today, an armadillo, and I took his picture.
No, I don't want that.
Yeah, leprosy.
Oh, that's right. He gets leprosy.
Yeah, armadillos, leprosy.
Armadillo. Leprosy.
Oh dear, I just got hungry. Got time. I want me to delete. Oh, you can delete a choice. That's what. There was one question on Lab Quiz 5, which closed at midnight last night. So your answers are open today. Well, there was one question there, it was a little ambiguous. And the question was, if you have E. coli grown on a Petri plate that was made by the 4-plate technique, why is it that some colonies on the surface are large and the colonies that are embedded in the agar are small?
Yeah, I put the facility, but it was.
The answer is because you generate more energy aerobically than anaerobically. But another choice was because E. coli are facultative anaerobes, and some people picked that answer, which would be correct. But the textbook author actually wrote, or the textbook provided that question. And actually what they're looking for is your understanding that facultative anaerobes can do both. But when they do it aerobically, they have more energy, they grow bigger. When they do it anaerobically, they have less energy, they're smaller. So the correct answer was because you generate more energy aerobic than anaerobically. And we talked about that. You ATP aerobically, you only get 2 anaerobically, so that's why the colonies are smaller. But that's what happens in 4-plate method with facultative anaerobes. So that one, I decided, okay, we have to resolve it, because both answers might be considered correct. So I texted Rich, because I didn't see a way to change it, and apparently he didn't either, so he said, we have to delete one of the choices. So we're gonna have to delete one of the choices, I guess, but I think what I actually asked him was just give everybody the correct, give everybody the points for it. But I think he's thinking about in the future, but in the future, I'm gonna re-upload all new tests, I have all new tests for next semester, so you're the last group to get these tests. I've been working like crazy. Okay, so back to this. So we did Koch's postulates, and when Dr. Koch was doing his postulates, remember he used solid nutrient auger, and he did the streak plate method, and why did he have to do solid auger and a streak plate method and spread them out instead of working from the broth?
Isolate it.
Yeah, he needed to isolate a pure culture of the disease-causing organism. Good, see how easy this is gonna be? Okie dokie. When you do an experiment, a scientific experiment, it's only considered valid if you have a control. If you don't have a control, you can't show that the variable that you changed is what caused the result. So, what's the purpose of using a control?
To compare.
To compare, it's your standard for comparison. So this is the standard thing that happens if I don't do anything to it. The experimental group, I'm going to change some variable and see if that results in something different from what my control group does. Good. So that's your control is your standard for comparison. Alrighty. We also talked a little bit about the subdisciplines of microbiology. In this class, I really focus on organisms. Not so much on structures like mitochondrial DNA and stuff like that, but I focus on pathogens. So the subdisciplines we looked at had to do with types of organisms that you study, right? So if you're studying bacteriology, you're studying